Hi, my name is Brenda Hutchinson. My partner is Ariel Allen, and this is our project on CRISPR and viral infections, focusing on using CRISPR to combat COVID-19 and/or influenza viruses. So, some abstract information. Viral infections are a pretty common occurrence. From the flu to more severe infections like COVID-19, viruses have shown to be notoriously challenging to treat, but there is a possibility that genome editing technology is the answer. This study aims to answer that question and dive deeper into using CRISPR technology as an alternative source to treat and or prevent these types of viral infections. We found that in studies involving CRISPR to treat influenza, CRISPR can reduce replication and initiate cell death within the virus. Using an RNA cleaving ability to select specific RNases, we can extract them from the virus, preventing the virus from replicating and or causing cell death, which would reduce the severity of the infection. So our hypothesis for this project is that we believe that CRISPR can be used as an alternative source to battle viral infections such as COVID-19 influenza, but is genome editing technology an option for a treatment, or could it cause an unwanted mutation? And how is the effectiveness of CRISPR as an antiviral determined? A brief introduction. For the past few years, the world has seen or faced an unprecedented challenge. The novel coronavirus, SARS-CoV-2 is its official name, but it's more commonly known as COVID-19, has completely flipped the world and the ways of life upside down. With supposed origins in China, COVID-19 spread to nearly every country, affecting tens of millions and taking the lives of 6.6 million individuals as of November 2022. A recent goal of the scientific community has been to develop treatments against COVID, but it has been challenging to do as viruses are harder to treat. At the pandemic's beginning, multiple studies explored the genome editing technology role in treating COVID by suppressing the replication of variants of interest. Similar studies have conducted research over CRISPR's potential to treat influenza A infections. We found that CRISPR is an excellent alternative to treating influenza, as CRISPR can reduce replication and overall reduce the effectiveness of the virus. Using CRISPR technology against COVID-19, we hope to show that it could be an option for treatment.
Some results that we found in the studies involves a case with mice and hamsters with mitigating influenza virus A and acute respiratory syndrome coronavirus 2. The scientists used CRISPR RNAs, otherwise known as CRRNAs, specific for PB1 and highly conserved regions of PB2 of influenza virus, and selected the CRRNAs that specifically target and reduce viral RNA levels in the cell. In mice, the Cas13a degraded influenza RNA in lung tissue efficiently. However, in hamsters, the Cas13a reduced SARS-CoV-2 replication and reduced negative symptoms of the virus. In our findings, we can conclude that Cas13a mediated targets of pathogenic viruses can combat respiratory infections involved with influenza and COVID-19.
An additional piece of results. The results demonstrated that the designed mRNAs express functional Cas13 proteins upon transfection of mRNA in A549 cells. LBU Cas13a localized to the cytosol, and LBU Cas13a and has additional nuclear distribution within the cell.
In figure 1, we can see the image that shows the apparatus for the mouse studies where they used an inhalable antiviral Cas13a mRNA. And in figure 2, we can see the infected mice that were dosed with the Cas13a mRNA. The red line shows the dosage that was given to the mice, and the black line shows the mice without the inhalable and viral Cas13a mRNA. Some methodology in rabbit reticulitis, LBU Cas13a and LBU Cas13a NLS, a variant with NLS sequence, are used to assist in transcribing mRNA sequences in vitro, and transcribed sequences were used to analyze and assess the RNA cleavage activity of the expressed RNases in conjunction with the guide RNA. Both variants contain a V5 epitope tag sequence. CrRNA and tRNA are derived from a genome segment of influenza virus A. Given the importance of SARS-CoV-2, a set of crRNAs that target highly conserved regions in the replicase and nucleocapsid regions of the genome were designed. The nucleocapsid sites would also allow targeting for any of the subgenomic RNAs produced by the virus. In this experiment, 9 guides were screened by the first transfecting viro E6 cells overnight with the cytoplasmic version of Cas13a and guides, and then infecting them with MOI approximately 0.1 of SARS-CoV-2. Prophylactic delivery was initially used because this virus exhibits very rapid kinetics in viro E6 cells, as well as the fact that they are interferon deficient. For conclusion, while studying influenza, this study revealed that CRISPR technology could effectively reduce replication and initiate cell death in viral infections. This was determined by using mRNA to examine the RNA cleaving ability of selected RNases. When examining the novel coronavirus, the effectiveness of CRISPR was determined by designing highly specialized and conserved cRNA molecules that target specific areas of the genome. And that's it. We have our references here. And my name is Ariel Allen, and this was our presentation over CRISPR and viral infections.